All right, so we'll talk about a ketogenic diet in our creatine transport deficient mice. We always want to start with the most important slide. That's the acknowledgements for all the people who have worked on this and helped fund this throughout the years. My lab, the Cincinnati Children's Research Foundation Animal Behavioral Corps, the Division of Neurology, and all of our funding sources, the ACD, NIH, Ultragenic, CEA, and Lumos. Very thankful to all of them. We especially want to acknowledge Kayla Miles because most of this work is her dissertation work. She's defending in 2 weeks and is starting a fellowship at Research Now on September 1st. So figure we'll honor her by showing her dissertation work here. So I learned a long time ago when coaching sports that all good coaches steal. So I stole this figure from Olivier and just added a little annotations to it. So basically there's, as you'll see through most of the talks today, there's a 2-hurdle problem or a 5-hurdle problem, depending on how you want to phrase it, in treating CTD. Our first hurdle is that the creatine must cross the blood-brain barrier. And then the second hurdle is the creatine must get in the right cells. So this is our main problem that we're facing as far as developing therapeutics. However, it does seem to be a little bit more complicated. So here on the left is data from my lab from a publication in 2018 where we knocked out the creatine transporter only in neuron-specific or only in neuronal brain-specific tissues. And you can see the significant reduction of creatine in the brain. The heart remains the same. Compared with wild-type animals, which are in blue. Over here on the right, we have a blood-brain specific knockout of the creatine transporter. And you can see here that the creatine levels in the brain are very similar between these two groups, which suggests that the primary source of creatine in the brain may not be through the blood-brain barrier, through the cells that make up the blood-brain barrier, the capillary cells that make up the blood-brain barrier, that there might be another system at play there. Just adding to the complications of this puzzle here. And really, we have a 3-hurdle problem, right? So not only do we have to cross the blood-brain barrier, not only do we have to get into the right cells, but we also have to have something that acts like creatine once it gets into the cells. And this is something that I think Alois and others brought up very well, is that it has to participate in the creatine phosphocreatine shuttle. And once it does, once it forms that phosphocreatine or phosphocreatine analog, it has to be willing to donate that phosphate group to ADP to make ATP. So we have a series of difficult issues there that have to be faced with any sort of therapeutic. So while all this is valuable, crossing all these hurdles are very valuable, it's going to be very time consuming. And in the interim, could we also look at other targets that could be disrupted by the lack of creatine? Because more than likely, creatine isn't the main action molecule for what causes these deficits, right? So primarily what we think is that it's the ATP deficits that are associated with creatine deficiency that probably lead to the phenotype observed. And we've shown in our knockout mice that ATP levels are reduced in the brain, heart, and muscle of these mice. This was data from Marla Perna back in 2016. So, and then we were able to follow that up with Alois's group and show that these brain ATP deficits actually are regionally based as well. With, if you look at the middle row there, you can see in the vehicle-treated animals, significant ATP reductions in the striatum, hippocampus. These are two tissues within the brain that are very important in learning and memory. The olfactory bulb has a significant reduction in the cerebellum. So these ATP deficits do seem like they're a reasonable choice for targeting treatments. So our overall hypothesis when we come to this study is that the lack of the creatine transporter causes a lack of creatine, a lack of phosphocreatine, and reductions in ATP, and this leads to our cognitive deficits. So what we wanted to try to do in these groups of studies was to see increasing ATP could improve the cognitive deficits. Well, how do we do this? So increasing ATP is not something that's just, there's not a lot of easy answers for this. And one of the things that we looked at doing was using the ketogenic diet. Most people are familiar with the ketogenic diet as a weight loss type of diet. So it's an Atkins style diet. It's a high fat, low carbohydrate diet. It stimulates the production of ketone bodies, which then can be used by the mitochondria to increase ATP content. And this increased ATP content has also been shown to protect against protect the hippocampus against mitochondrial poisons. So think about reducing ATP because of the lack of mitochondrial output. The ketogenic diet's been used to treat a variety of refractory epilepsies. And it's been proposed for use in several neurodegenerative diseases that have been linked in disrupted energy production. Actually, another study that our lab does is we look at mitochondrial function in platelets from humans that have mild cognitive impairment. And a colleague of ours uses different behavioral or dietary strategies to see if we can reverse that. So it's become widely accepted as one of the ways that you can mind the gap, so to speak, of energy deficits and cognitive performance. So jumping right into what we've done here with the ketogenic diets, we started our wild type and knockout mice on a ketogenic diet or a textured match control diet on postnatal day 21. The ketogenic diet is 75% fat, 9% protein, 3% carbohydrates. The control diet was 7% fat, 18% protein, and 63% carbohydrate. So the reason that we do a textured match is you can't get a 75% fat diet to look like normal everyday mouse chow. So they have to use more, it's more of a Play-Doh type of consistency. And we wanted to make sure that the animals were consuming the same amount and that they just didn't like the diet or whatever else. We also decided to start the mice on postnatal day 21 to mimic an early life start on the diet. Sometimes modeling when you're going to start a treatment is difficult. Mice and humans obviously don't age at the same rate. And postnatal day 21 is a good time to start because this is when mice are weaned from the dam and can then eat on their own. So this gives us the best chance at an early life start on the diet. The mice were kept on the diet for about 40 days. So we started behavioral testing on P60, which is considered young adulthood. And we also examined neuronal morphology in a separate group of mice. Okay, so the first thing we wanted to make sure was that the ketogenic diet worked. So we measured blood glucose, beta-hydroxybutyrate, which is the main ketone body. So when you go into ketosis through a ketogenic diet, this is what's actually elevated, which you can measure. So the animals on the ketogenic diet had reduced blood glucose. That's both in wild type and knockout mice. Whereas the control diet animals' blood glucose were still elevated. They also showed increased plasma beta-hydroxybutyrate levels on days 31, so 10 days after they started the diet, and when they were going into the diet, even though there was a little dip in the knockout mice here. One of the things that we did notice was that the ketogenic diet stunted the growth of both groups. And this is a fairly well-known phenomenon when it comes to mice. So we were sort of expecting that, but you can see that the red and the yellow lines there are the animals on the ketogenic diet, and that they have reduced body weight throughout testing. So one of the side effects that we weren't expecting was that the ketogenic diet actually ended up severely increasing the mortality of our creatine transport knockout mice. So this is a survival curve, a standard survival curve, and you can see in the yellow line here, this is the knockout mice on the ketogenic diet. And by the time that they got to behavioral testing, about half of them had survived. And then the wild type mice, we still see a reduction in survival of those as well. We did see some change in the control diet survival. Usually these animals, we don't have any issues with at all. It could have been a host of things that have gone on there. But the take home here is that the the ketogenic diet did have negative effects on the animal survival. So then we also looked at, so then we began our behavioral testing. The first test that we always do is to check the activity levels in the animals. This is just an open field arena with infrared beams that measure how many times the animals ambulate throughout the arena. We were able to see that the knockout mice on a normal diet were hyperactive in the first half hour of testing. That's a fairly consistent finding between those two groups. But the ketogenic wild type animals and knockout animals had severe hypoactivity compared with the normal diet groups. There was a main effective diet there that, regardless of the gene, showed this hypoactivity. All right, so the next test that we generally do, the animation got messed up, so that's fine. Okay, this is the Morse water maze. So the main, we'll go down to this one. The main thing on the Morse water maze is that we put the animal here. Its job is to find the platform here. They can't see this platform if it's submerged underwater. We measure a whole host of things, but the best measure that we have right now is something called path efficiency. If we draw a straight line between the X and the circle, we then take the animal's path, which is the white up here, and we calculate the difference in distance between the straight line and the path the animal takes. Our knockout animals show their typical, so this is the lower the score, the worse. Our knockout animals show their typical reduction in path efficiency compared with wild types. So the green line versus the black. Again, this is acquisition and this is reversal. The big difference here is we just move the platforms around to test the cognitive flexibility. Interestingly, the ketogenic diet animals, regardless of genotype, also show deficits in both measures of this, suggesting that early life start to a ketogenic diet leads to learning deficits. And we actually published the wild type data earlier this year to show this effect of this model. Novel object recognition, pretty standard test. We've talked about it plenty. Expose the animals to 2 familiar objects, wait an hour, and then switch one of the objects. If the animal remembers it, they should spend more time with it. This is one of the rare times we did not replicate our pretty standard novel object effect. The reasons into why, I'm not absolutely sure, but this is in the normal diet. You see no difference here, same with the ketogenic diet. And then we did conditioned fear. This is a test where we expose the animals to a tone and a foot shock. Ideally, they're going to remember that they were shocked when they were placed, when they heard the tone and in this arena. Day 2, we see if they're scared of the arena, if they remember the arenas where they got shocked. And the response to fear is freezing. And you can see that we were able to replicate our common finding of contextual fear deficits in our animals. We also were not able to alleviate this with the ketogenic diet. On day 3, we changed the floor to give them a different environment. Then we exposed them to the tone and see how long they freeze. There was a main effective gene, so we did replicate our effect showing a acute fear difference as well. And again, no effect of the diet. So after all this, we also wanted to look at changes in dendritic morphology in the hippocampus. The hippocampus is the primary region that's involved in spatial learning and memory. There's inputs from most major brain regions. And we specifically looked at these neurons up here. So this is just our mice, our knockout mice crossed with a mouse that has a green fluorescent protein that's expressed in a small subset of hippocampal neurons. So these are wild type mice on this side. These are knockout mice on this side. Looking at these, you can't tell a whole lot of difference. So we do what's called a shoal analysis. This is where we just draw circles in increasing size from the cell body, just count the times that the dendrites cross these circles. In the first study that we did, we showed that the knockout mice have reduced crossings as they get further away from the soma, suggesting a lack of dendritic complexity, dendritic branching. And again, we were not able to alleviate this in our ketogenic diet tested mice and our treated mice. We were able to replicate the initial finding of the reduced intersections, and the ketogenic diet did not improve that. And this just shows the replication again. Okay. So then we wanted to, because our hypothesis was if we could elevate the ATP levels, we should be able to ameliorate the cognitive deficits. Because of the lethality of the diet, because of the lack of survival, we looked at ATP levels in younger mice. So we only put them on the diet for 2 weeks. And what we found here that's sort of interesting that we need to follow up on was that there's no difference between knockouts and wild types at P35 and ATP levels. Now the adult animals, is what I showed you before, those were all P60 or postnatal day 60 or older. But on postnatal day 35, we didn't see these deficits, and if we don't see the deficits, the ketogenic diet can't fix them. But we do see a small elevation. This isn't statistically significant. It'd be interesting to follow up on as the animals get older with a few different modifications. So I think the big question that we came away with when we finished the study, or as we did this study, is what's causing the decreased survival, the mortality in our mice? And during one of Kayla's committee meetings, it was brought up that, you know, what do the mice, when you go in the morning to feed the mice or whatever, and you find one deceased, what do they look like? And she says, I never found any. So we started wondering maybe if, because the creatine transported knockout mice are a little bit smaller than their cage mates, that there was a dominance issue and that the cage mates were, for lack of a better term, eating the other mice. So to test this, we took a group of mice and we just singly housed them with the diet. So they didn't have any cage mates. We usually house the mice, 4 mice to a cage. So GH is group housed, standard housing, and SH is singly housed. So you can see the red and the purple here, and these are only knockout mice, that when we put the animals in by themselves, we see a significant reduction in the mortality, which would sort of validate that it's something that the cage mates are doing to the animals. The weight didn't change between them. So you can kind of say that it wasn't due to a lot of weight issues. Actually, the weights did increase, which again could be some dominance issues or something like that. And the take home from that is that we believe that the stronger wild type mice cannibalize the weaker knockout mice. The fun of animal research sometimes. So for summary, the ketogenic diet didn't improve cognitive performance in our knockout mice. It did lead to an increase in mortality, but that was likely due to housing conditions and not the diet itself. The reduced dendritic arborization that we see in our knockout mice was not improved by the ketogenic diet. And I will say that I do believe that most of the animals that were put in the ketogenic diet for the arborization study were singly housed. And there are no ATP differences between the mice, and that could be due to the age of tissue collection. The conclusions, it's not starting the ketogenic diet in very young mice is not an effective treatment for creatine transporter deficiency. We're going to try starting the treatment in older mice. We also will probably single house the mice to avoid the dominance-based cannibalization, and ATP levels could be evaluated at several ages. And we will finish with, again, the most important slide, thanking everyone who did this and was involved in all of these studies, and we'll be happy to answer any questions after Ultragenyx does their presentation.
Great. Thank you so much for that, Dr. Skelton. We will come back to you for questions after Dr. Ayalon's talks. And just a brief introduction. Let me see if I can, there we go. Okay,